In this video, I'm just going to quickly go through the autosomal recessive, autosomal dominant practice questions that we put up. You'll notice a lot of them are actually taken from National 5 level course, but they are the tricky ones from National 5. They should give us a little hang of the autosomal dominant and recessive conditions. Not all of the questions, because they're from National 5, are related to humans, but they are still applicable. So if you haven't already completed them, pause between the questions before I go through the explanation. So we're going to go through question one, pause if you haven't done it already. Okay, so from the family tree, in which generations are all dogs heterozygous, not is? Are all dogs heterozygous for home coat colour? So the way we do this, I've written these out. So we know that heterozygotes have a dominant and a recessive allele. We know we're not looking at sex-linked at the moment. So a heterozygote will show the dominant phenotype. This rules out the parental because one of the individuals does not have the dominant phenotype, so they can't be a heterozygote. We know that the individual here, from looking at the family tree, the pedigree, we know that that's likely to be the recessive phenotype. So they will be homozygous recessive genotype. We don't know what the allele on this one is. All we know is that it's not the parental is not heterozygote. So looking through this, we know this side of the Punnett square if it was important, and we know this side of the Punnett square. For the F1, is it possible that these are heterozygotes? So all of the F1 seem to have the dominant phenotype. We know that the black square is dominant due to the fact that in the parental generation, two differently phenotyped individuals were crossed, and that the black is all that we see in the F1. If it was the other way around, the white would have been a homozygote, and we would have expected, so the white would have been the dominant phenotype, and we would have expected some white offspring in the F1. So the fact that the parental generation has two differently phenotyped individuals that were crossed, and all we see in the black, this tells us that it's very unlikely that the black isn't the dominant allele. So as the small t parent can only pass on the recessive allele, and all the F1s are black squares, they must have a small t, but they must also have a big T in order for them to have this phenotype. So F1 would seem to be heterozygotes. So as the TT can only pass on the recessive allele, and all the F1 are black squares, they must be heterozygotes. If the F2 were looked at, we don't know that they're not heterozygotes. It is possible, but it's very unlikely if we're crossing the heterozygote with either a heterozygote or a homozygous dominant here. It's unlikely that that would give you only heterozygotes. So that means the answer to that one is just the F1. For the next one, we're looking at a hairy-stemmed pea plant. It's crossed with a smooth-stemmed pea plant. All the F1 have hairy stems. What is the genotype of the F1 plants? So for this one, again, we've done a wort example here. So any time in autosomal inheritance, two differently phenotyped ones are crossed, one of them has to be homozygous recessive, because in autosomal inheritance, for you to have a different phenotype, you must be homozygous recessive. Because if you had one of the dominant alleles, if you're either heterozygote or homozygous dominant, you would have the dominant phenotype. So that means one of these is the homozygous recessive. It means that all of the F1s will either be heterozygous or homozygous recessive, as it isn't possible for them to be homozygous dominant as one of the parents doesn't have that allele. The fact that all the F1s are hairy, which is the dominant phenotype, they then must be heterozygotes, because again, like in the previous question, all of the F1 plants are showing the dominant phenotype, and one of the parents can only give out recessive alleles. So it is likely that they are the heterozygotes. So heterozygotes. For the third question, so whatever that is a dominant characteristic in humans which causes the person to have two rows of eyelashes, so double eyelashes. A woman who is homozygous for the condition. Now let's have a look. It's dominant, so she's homozygous dominant. and a man who is unaffected. So if he doesn't have a dominant condition, it means he's homozygous recessive. What proportion of the children will be expected to have this? So you can see the thought process. She is homozygous dominant, he is homozygous recessive. You could, if you get good at this, be able to just snap your fingers and say what the answer to that one is. So we know that she is homozygous dominant. So this is how our Punnett square starts. And we know that he is homozygous recessive. So we can complete the Punnett square as here. So all of the children will have the disorder because the female will pass the dominant allele, which gives the condition, to every one of their children. So actually, his genotype is completely irrelevant to the question. As soon as she was homozygous dominant, we knew that all of the children will have the disorder. For the next one, a hairy-stemmed pea plant was crossed with a smooth-stemmed pea plant, and all of the F1 had hairy stems. The genotype of the hairy parent. Now, as we said before, if two autosomally controlled phenotypes are crossed, then one of them must be the homozygous recessive, or else both of them would have the same phenotype. The other we don't know, except that they must have at least one of the dominant alleles. So we don't know for one of the parents, but they must have at least one dominant allele, like that. So that's what we know so far. It is likely that because all of the offspring were hairy and there were no recessives, so if that was, for example, like that, you would expect at least 50% of the children to have the recessive phenotype, these ones here. But because they don't, it is likely that that parent, whose genotype we don't fully know, was probably homozygous dominant, because these are the two options. You can have this one, or you can have this one. It's likely that we've got the top example, the big T, because otherwise we'd expect a 50-50 split. Again, it's a bit unsatisfying to answer these questions based on the likelihood rather than knowing for sure, but it is probably the answer that they are homozygous dominant. And that's what the SQE think the answer is. So there we go. In humans, the allele for free earlobes is dominant to the allele for fixed earlobes. So capital E for free, lowercase e for fixed. The diagram shows some inheritance. So without even reading the question, we know that if someone has fixed earlobes, they must be homozygous recessive, because if they had even one of the dominant alleles, they would have the free earlobes. This is what happens if you click on the question. It's quite annoying. So, which table correctly identifies the genotypes of the parents? So, the one that has fixed earlobes must be recessive. So that is the P square. So that's the question. So either A or B is going to be our answer for this one. And again, the worked example is here. So P has to have fixed earlobes. That means they're homozygous recessive. So we're left with A or B. Q has free earlobes, which is the dominant phenotype. We see that it is able to mate with another individual that has the dominant phenotype and produce the recessive phenotype. So this one has dominant phenotype. We know it is the heterozygote, actually, because it has one parent who is homozygous recessive, and then it has the dominant phenotype. So it must have inherited one recessive allele, and yet it still has the dominant phenotype, so it must be big T, small t. So big E, small e, which means the answer is B. Once you get good at these little tricks, it's actually pretty easy. We'll say we don't know these answers because we're cleverer than you as teachers. It's because we've done them 1,000 times. In gerbils, a agouti colour is dominant to white. Some heterozygous gerbils, so immediately you're thinking big letter, small letter. So two of them seem to be crossed. 56, I shouldn't click on them, should I? 56 offspring were produced. What was the expected number of agouti gerbils? So you've got two heterozygotes that you're crossing. So if we remember our numbers, 1 in 4 times you'll get the homozygote, 2 in 4 times you'll get the heterozygote, and only 1 in 4 times will you get an offspring that is not going to have the agouti phenotype because it is a homozygote. So the answer is 42 because of what I've just said. So 1 in 4 times you'll get one that's not agouti. That is 3/4 of 75%. So 3/4 of the 54 will be agouti, which is 42 gerbils. In fruit flies, Drosophila, the allele for normal wings is dominant to the allele for short. A normal wing was crossed with a short wing, so we know one of them's homozygous recessive, and it's the short-winged one. All of the F1 had normal wings. If the F1 were to mate, what's the percentage of the F2 that would be expected to have normal wings? So we know that the parental generation has one who has the dominant allele, and we don't really know what the other allele it has is, and we know that its other parent is homozygous recessive. So we know that all of the F1 produced will be heterozygotes. So because they're all normal winged, they got a capital N from their normal and a smaller case n for so they haven't told us what the thing is, but yeah, cool. And a smaller case n for the short wing parent. So all the F1 are that. If we plug it into our normal Punnett square, we have 1 in 4 chance of homozygous dominant, 1 in 4 chance of homozygous recessive, and a 2 in 4 chance of heterozygotes. We want to know the percentage of the F2 with normal wings. It's the same as question 6, but in reverse, basically. So 3/4 or 75% will have the dominant phenotype. So that was easy, wasn't it? So 3 of the 4 F2, so just probably put the word F2 there rather than P. There we go. 3 of the 4 F2 will have the normal, and 1 in 4 won't, so it'll be 3/4, or 3:4 ratio. The diagram below shows the section of a matching chromosome found in 4 fruit flies A, B, C, and D. The genes are here, which is homozygous for both genes. So you're looking for one where both of the squares here and the squares here are matching each other. So that matches, but that doesn't. That doesn't match, but that does. That doesn't match, that doesn't match. Hey, look for D, it matches. Yeah? And then it looks like I was just going to wing the rest of it, apparently. I'm sure we can do this quickly. So in corn, yellow seed is dominant to purple seed. So this cob shows a few yellows and a few purples. The seeds have been counted. So each of the seeds is an independent individual. The genotype of the parents of the cobs. So if you are producing an equal number of phenotypically dominant and recessive, we can walk this through in our head. So the easiest way to do that is if one of the parents is homozygous recessive. Because if one of the parents is homozygous recessive, we can actually completely ignore their genotype. Because either they are going to have an allele given to them by the other parent who gives them a dominant allele, and then they're going to produce the dominantly phenotyped child, or the other parent gives a recessive allele, and then they produce a recessively phenotyped child. So if we cross, in this case, when the offspring give a 50-50 ratio, it usually means, see if I'm right, I'm right, that you've crossed a homozygous recessive with a heterozygote, because we can ignore this parent's genotype completely. This one will 50% of the time give the dominant allele and 50% of the time give the recessive, and that's what we see in the phenotypic ratio of the offspring. Which of the following is discrete variation? Not something we recover at the higher course much, but it's good to remember. Discrete variation is single gene variation, usually. It's an either or phenotype. So earlobes attached or unattached is either or. Height can be any height, you can have any hand span, and you can have any leaf length. It's not an either or. What percentage of students have green eyes? It's 160 students. Green eyes is 24. If you add that up, you get 160, I imagine. So 24 over 160 times 100 gives you 15%. And the last little bit, we'll power through this. So the possible genotypes for the F2. So big N, big N, big N, small n, and small n are the 3 possible phenotypes. If you write an extra big N, small n in, it doesn't matter. The phenotype of parent 1. So parent 1 looks like it has normal. Parent 2 looks like it has the recessive phenotype, which is The normal phenotype. Parent 1 would be polydactyl or have extra fingers, and parent 2 would have the recessive phenotype, which is normal. In the F1, all of the F1 have a dominant and a recessive allele, so will be polydactyl. Why is that the actual phenotypic ratio of the F2 can often be different to the predicted? It's because the choice of allele during meiosis is random. So we say the variation produced is random from sex. Hopefully that was useful. If you have any questions, please do ask.